In this question, you have to identify the vector for introducing a DNA fragment in human lymphocytes. Option A, retrovirus. Actually, retrovirus are commonly used as vectors for introducing DNA fragments in human lymphocytes. Lymphocyte from blood of patient are grown in culture outside the body. Then a functional gene is introduced by using a retroviral vector into these lymphocytes. Hence, it is the correct option. Option B, TI plasmid. It is incorrect because TI plasmids are used for genetic transformation in plants and not human cells. Option C, PBR322. It is also incorrect because it is a plasmid vector which is used for cloning in bacterial systems like E. coli. Option D, phage. It is also incorrect because phages or bacteriophages are used as vectors for bacterial system.